Hi Year 12s and more 11s and 12s. Continuing on with week 2 of cells, which is the IBSL content. We're moving on to prokaryotic cells, and this is another shortish video. Let's go straight into it, talking about what is the structure of a prokaryotic cell. And there's two types of cells. You learned this in Year 10, probably Year 9, Year 8 as well. Eukaryotic cells and prokaryotic cells. Now we're going to focus on eukaryotic cells in another video. In this PowerPoint we're talking about just prokaryotic cells. Now prokaryotic cells are going to be unicellular organisms, so one-celled organisms. They generally don't have a nucleus or any internal compartments. So what's special about a prokaryotic cell is they do not have a nucleus or an internal compartment. So no organelles essentially. Except for ribosomes. They do have ribosomes. So it's better to say they don't have membrane-bound organelles, because ribosomes aren't membrane-bound. So, essentially when we're talking about prokaryotics and unicellular organisms, all prokaryotic cells are unicellular organisms, but not all unicellular organisms are going to be prokaryotic, because you can have some eukaryotic unicellular organisms too. They will have a nucleus, they will have internal compartments. It's only prokaryotic cells that don't have that nucleus or internal compartments. From the evidence that we have and from the research that scientists have done, we do believe that prokaryotic cells were the first organisms to evolve on Earth, and because of that we still foresee they have the very simplest structure of any of the organisms we've discovered. Very small in size, prokaryotic cells are found virtually everywhere, because we're talking about things like bacteria. You have bacteria in your body, you have bacteria in your skin, there's bacteria in your desk, there's probably bacteria on my microphone, on my computer screen, on your computer screen. There's prokaryotic cells everywhere. Like all cells, they have a cell membrane. Prokaryotic cells, on the other hand, also have a cell wall, which is outside of the cell membrane. It's thicker and stronger than a membrane. They don't have any skeletons or protection like we do, so they need to have that cell wall to help provide structure for the cell. Now, I'm not sure you can see very clearly on the PowerPoint. You might need to zoom in or make the video a bit bigger. And it's important, especially when you're drawing in the IB, that you make the cell wall look different to the cell membrane. So you can see here I've done a very thin line for the cell membrane, where there's a very thick line for the cell wall. Now what you could do is actually do double lines for the cell wall and a single line for cell membrane if it's a bit harder in an exam or a test to draw like that. But you need to be able to visibly see the difference. And the cell wall is thicker, so you either use a thicker line or you draw a double line for the cell wall. The whole idea of the cell wall, like I said before, basically provides structure to the cell, prevents the cell from bursting. On the inside of the cell, like with the eukaryotic cell and all types of cells, the entire interior is filled up with cytoplasm. The only organelles that they have is ribosomes. Like I said before, they're not membrane bound, so that's how we get away with saying that prokaryotic cells have ribosomes. You will see when you look at them under an electron microscope, and I'll show you that a little bit later, maybe when you're back in class, there's a section of a prokaryotic cell that it appears lighter. Ignore everything falling over. It appears lighter, okay? And in those sections that are lighter, that's because that's where the DNA of the cell is. Now, if we were to actually see that properly without probably with an electron microscope, for example, what you'd see is that this is actually the nucleoid. The nucleoid region is where the DNA is stored. It's kind of just like a big ball of entwined DNA there. It's generally one circular DNA molecule, but it's all entwined. And that's how we draw it, almost like a squiggly line when we're drawing this. Now, it is important that you are able to draw prokaryotic cells, so I would recommend copying the one that I've got here on the right. Is a nice hand-drawn image by me on my iPad. There are some different features of the prokaryotic cell that we haven't talked about in the slide that are in here that you probably need to know as well. So we've talked about cell membrane, we've talked about cell wall, we've talked about the cytoplasm. Ribosomes are the site of protein synthesis. We'll talk about them a lot more in detail. We talked about the DNA being found in the nucleoid. There is this different section down here. It's just like a little circle here called a DNA plasmid. The idea of that DNA plasmid is it's a section that bacteria or other prokaryotic cells can exchange genetic information, and that way they can learn new tricks. And we'll talk about this a lot more in detail later on in the course. But for example, when you have superbugs or antibiotic-resistant bacteria, a lot of that's through the information that's shared through the DNA plasmid. Humans have also grabbed the DNA plasmid, and we've utilised that technology. A lot of genetic engineering, for example, how we manufacture insulin, is all through the use of DNA plasmids in bacteria. You also have, over here on the diagram, you've got some pili. Now, pili are very fine hairs that are all around the entire prokaryotic cell. Basically, they help them to stick to things, help trap objects, etc. We also have a flagella drawn on here. Now, not all prokaryotic cells will have a flagella. The idea of the flagella, it's a whip-like structure at the end of the prokaryotic cell, and basically helps with movement. So if you're a prokaryotic cell that's not moving, you probably don't have a flagella. If you do need to move throughout, for example, liquid, etc., flagella will be used almost like a propeller to propel the prokaryotic cell forward. Keeping on with the topic, moving into cell division in prokaryotic cells. All living organisms, we know all cells come from pre-existing cells. We learned that in cell theory the other week. So how that happens is through cell division. Now, in prokaryotic cells, it's a very specific type of cell division, and we're looking at binary fission. Binary fission is a type of asexual reproduction. What happens is the single circular chromosome is replicated, then you've got 2 copies of the chromosome. Generally, they'll attach to the cell membrane, and as the cell starts to elongate and grow, those 2 chromosomes will be pulled to either end of the cell. Once the cell gets too big, you start to have division in the cytoplasm, and called a cleavage furrow, happening in the middle of the cell, and then 2 daughter cells are created, indicating that each daughter cell will have one of those 2 copies of the chromosome. There are some videos here showing this. So look, this one. We'll start with this one here. This is an artistic rendition of what's happening in cell division here. So you can see the chromosome doubles, the cell elongates, divides into 2, and you have 2 identical cells at the end, the same as the first cell. This is more of you can't see obviously the chromosomes in the middle. This is a sped up image of what's actually happening in real life. You have some rod-shaped bacteria over here, and they're multiplying really fast, pretty much doubling, because they're going through cell division, and you can see how fast that is actually, obviously it's sped up, but you can see how fast the population of prokaryotic cells would grow in that instance.